9 a.m. my time, Central Time. It's probably 4 p.m. in Europe, in France. So good morning, everyone, or good afternoon, wherever you are in the world. Thanks very much for joining us to this 5th webinar offered by the group Polyphenols. The group Polyphenol Society is an international association founded in France in 1972 with the aim of promoting research on plant polyphenols from their most basic and fundamental biophysical chemical properties to their most diverse applications in food and agricultural, pharmaceutical, and cosmetic sciences and technologies. The group Polyphenols started to develop this series of webinars in Polyphenols Research in 2022 with presentations about polyphenols in soil systems, food and beverages, and about their characterization. If you didn't have the chance to participate in the previous webinars, you can have access to the recordings on the website of the group Polyphenols. I'm Dr. Aude Watrelot, an assistant professor and extension specialist of phenology at Iowa State University, focusing on polyphenols' chemical properties in wines. I'm the vice president of the group Polyphenols, and I'm co-hosting the webinar today with Dr. Panagiotis Arapitsas. Before I give the floor to Dr. Arapitsas, I would like to provide you some instruction. So as I mentioned, the webinar is going to be recorded and will be available on YouTube in about a week. Please type any questions you may have during the presentation using the chat box that you might find at the bottom of your screen by a click on the 3 dots. Your questions will be answered at the end of the webinar. At the end of the webinar as well, you will receive an email to complete a Qualtrics survey about how we did today and what we will need to change for future webinars. So please just take a few minutes to complete it. Also, if you need a certificate of participation to this webinar, please send me an email to request it, and then you will receive it in about a month. So Dr. Arapitsas, the floor is yours to introduce yourself and our speakers today.
Okay, perfect. Thanks, Aude. Hello to everyone. Welcome to this webinar. So yes, my name is Panagiotis Arapitsas. I'm professor in enology in the University of West Attica in Athens, and also researcher in Fondation Edmund Mach. And it is a great pleasure for me today to introduce you Catherine Dadmun. She has a very interesting background because she studied initially both chemistry and French, and these two combinations drove her in study enology and wine chemistry. So she got her degree, her master's degree at the Cornell University in food science and technology in 2020. And at that time, she started to work with anthocyanins in interspecific hybrid grapes. And then she started her PhD focused on that while moving between the Cornell University in US and the University of Burgundy in France, trying to finish her study. She's finishing in next fall. She will defend her dissertation hopefully this fall. I wish her my best luck for that. And she also like a lot the science communication, so I think she will be an excellent presenter for this webinar. And the title of her presentation is Unique Polyphenolic Profile of Disease-Resistant Hybrid Grapes and Wines. Catherine, the floor is yours.
Thank you. Okay, so I'll share my screen. Hopefully that's correct. And I'll bring my camera over. Okay. So hello. Thank you so much for that introduction. As he said, my name is Catherine Dadmun, and my PhD dissertation research has been a collaboration between Cornell University in the United States and University of Burgundy-Franche-Comté in Dijon, France. And I'd like to thank the Group Polyphenol Committee for inviting me today to discuss how metabolomics can be used to explore the unique phenolic profile of disease-resistant grapes and wine. So let's see if this works properly. Okay, we'll see if that works. So even if you like to drink wine, you might not be very familiar with interspecific hybrid cultivars. These go by a number of different names all over the world, varied between, oh, my clicker's not working. just hybrids. PIWI is a German abbreviation for fungus-resistant grape varieties, disease-resistant cultivars or varieties, and non-traditional cultivars. And when I say the term cultivar or varieties, what I'm talking about are the grape types. So you know a lot of them probably if you drink wine, like Chardonnay, Merlot, Pinot Noir are all different varieties. And the three that I just listed are all from the species Vitis vinifera, which is known as the traditional European wine grape. The interspecific hybrids are a cross of Vitis vinifera and other Vitis species. For example, Chardonnay is a cross between Céval and Chardonnay. Chardonnay is a cross between Pinot Noir and Gouais Blanc. And as you can see here, Céval is a cross of a number of iterations of Vitis vinifera, as well as other Vitis species. And many people will often ask me, their first question is, are these hybrids GMO or genetically modified organisms? And the answer is no, these are traditionally bred through select crossings, and sometimes genetic markers are used to find selective attributes, but they are not GMO. So when defining what these selective attributes are, there are two main goals. The first being disease resistance and cold hardiness. Vitis vinifera is a relatively susceptible grapevine, and disease resistance and cold hardiness to pathogens and vineyard diseases is one of the main goals of designing and breeding these interspecific hybrid cultivars. But the second parameter, which is quite imperative, are quality parameters. So aroma, taste, color, and tannin are all really critical parts of designing interspecific hybrids, because originally hybrids developed way back in the 1800s were not developed with quality in mind and had low quality parents. And this negative stigma can still carry on today despite modern research in creating high quality hybrid cultivars. So both the wine research world and the wine industry itself are finding stakes in interspecific hybrid cultivars due to the pressures of climate change. So climate change can mean more than just hotter weather. It can mean severe early or sporadic frosts. It can mean more inclement rain and an increased number of severe weather events. And all of these things can increase disease pressure in the vineyards. There's also a critical push worldwide to significantly reduce pesticide use in vineyards. Specifically in the European Union, there's a goal to reduce pesticide use by 50% by 2030. The OIV, the International Organization of Vine and Wine, is on board with this. In their most recent strategic plan, the first axis was to promote environmentally friendly vitiviniculture. And as a pillar within this axis is to promote intra and intervarietal diversification in commercial vineyards. And the data has shown that disease-resistant cultivars do demonstrate a massive reduction in the need for pesticide and fungicide use. So they show a lot of potential. Additionally, the wine industry has expanded globally to areas that previously were considered not well suited for wine grape cultivation, and in part, in some places around the world, is thanks to use of these hybrids. But there's an overall global expansion of wine production. So to summarize, interspecific hybrids show a lot of potential due to the impact of current and future climate change, the global expansion of the wine industry, and the recent research development of quality of hybrid cultivars. So overall, my research begins to explore how to characterize the chemical profile of interspecific hybrids and particularly observe how compounds interact in this matrix. On the other side is understanding sensory perception of interspecific hybrid wines, particularly in the realm of general consumers, quality perception, and color. And overall, the goal is to optimize winemaking and aging strategies. And naturally, as is in a lot of different research, are intertwined and overlapped. So what role can metabolomics play in exploring these interspecific hybrids? Many of you attending today are probably aware of what metabolomics is, but I think to truly explore the potential of metabolomics, it's helpful to clarify the root of the term. So naturally, metabolomics is a study of the metabolome, which is the set of metabolites in an organism. Metabolites are small molecules that take part in cell metabolism, and as you likely remember from Biology 101, metabolism is a series of life-sustaining chemical reactions in organisms. So metabolomics is the study of this metabolome, which is this massive network of interlocking metabolites. And generally, there are two approaches to metabolomics. The first being non-targeted, which looks at an overall fingerprint of the entire metabolomic network. And this is often used for discovery, can generate hypotheses and be very exploratory, and it's pretty much accepted and a given that when you're looking at this massive network, you will have a large proportion of unknown metabolites that are unannotated or unidentified. But it gives you a big picture look at the metabolome. Then there is a targeted metabolomics, which is looking at a selected subset of known metabolites. And in the case of my research, that subset is polyphenols. So many of you probably know this tree of the polyphenol family like the back of your hand. And in the context of wine, I just wanted to clarify two small things. The first is that in my study of color perception, the targeted family I look at the most is anthocyanins because these create color in grapes and wine. Excuse me, I have a bit of a cold, so. The second thing in wine is that when you attend a wine tasting, you will likely hear talk of tannins because they are important organoleptic properties of wine that create mouthfeel and body. And in the polyphenol family, there's actually two types of tannins. There are oligomeric forms of flavin-threols, which are known as either condensed tannins or proanthocyanidins, and these are extracted from the grape and plant itself, such as the skins and the seeds. And then there are hydrolyzable tannins made up of elagitannins and gallotannins, and these are not found in Vitis vinifera grapes themselves. They have to be extracted from wood and usually oak during the winemaking process. So metabolomics can tell a story about this massive wine matrix. Particularly within non-targeted metabolomics, it's possible to discover unknown compounds, to compare entire chemical fingerprints even when 90% of the metabolites are unidentified. And with a targeted subset, you can define profiles of unstudied cultivars, such as interspecific hybrids. And particularly with targeted, you can quantify differences between, for example, winemaking treatments or different origins. Excuse me. Both of these approaches, especially when used together, can provide greater insight into production strategies, which have applications to the wine industry, sensory perception, which has applications to consumers, and overall can be a way to better understand this complex chemical matrix. Non-targeted metabolomics has been used to demonstrate the impact of terroir and vintage on a chemical fingerprint of wine. So terroir meaning the origin and environment that the wine comes from, and vintage meaning the year it was made. So here there were two distinct nearby vineyards from Côte de Nuit, CN, and Côte de Beaune, CB, in Burgundy that were compared using non-targeted metabolomics, and what they found was that they could differentiate the grapes and corresponding wines from these distinct vineyards according to their chemical complex fingerprint. And what notably, they found that vintage has a higher impact on metabolomic pattern than terroir, which has been seen in a number of different metabolomic studies. Non-targeted metabolomics can also uncover novel compounds. So in this study, there's a comparison between multiple different American Vitis species, Vitis vinifera, and a number of interspecific hybrids. And for the first time, they identified hydrolyzable tannins and their precursors in grapes of American Vitis species. As I mentioned, hydrolyzable tannins are not found in Vitis vinifera grapes, and they are desirable markers of wine quality and are extracted from oak because they can improve wine body. So this novel discovery can have implications on how breeding programs are designed in the future and the role metabolomics might play in hybridization to observe unique markers carried from parent to hybrid cultivar. Excuse me, I'm so sorry. I'm a little sick. So, thirdly, non-targeted metabolomics has been compared to sensory perception, which I have a lot of interest in. In this study, they were exploring if 3 different clones of Pinot Noir, 3 different types of the same grape, could be distinguished if vineyard location and winemaking practice were held constant. If so, if they could distinguish these 3 clones using non-targeted metabolomics, they wanted to see if they could understand the diversity of sensory attributes in the wine, and particularly if unrestricted metabolomics could link associations between certain metabolites and their sensory impact, aromatic and taste attributes. They did in fact find that one clone was distinct from the other 2, and they found that this 3rd clone had higher scores for vegetal and herbal aromas, while the other 2 clones had higher scores for caramelized, stone fruit, and red berry aromas. Particularly, they found that secondary metabolites and their concentration, namely phenolic compounds, were major factors in determining this distinction of the 3rd clone. And these sets of metabolites within the phenolic family can have huge implications on organoleptic perception. So I thought there was a really excellent quote from this article that gave a good representation of the benefits of metabolomics. So they say that wine is a very complex matrix, and the level of individual the level of individual metabolites are not necessarily as important as their interaction with other compounds. This is why the use of metabolomics can allow a holistic perspective in monitoring several aspects of wine quality. So I think that connection of non-targeted metabolomics and sensory perception is really fascinating. Now, in the case of the metabolomic profile of disease-resistant varieties, in this study, they compared 17 different disease-tolerant varieties or disease-resistant varieties alongside 6 established high-quality Vitis vinifera varieties. And using 116 different volatile and non-volatile metabolite variables, they could map here in green the disease-tolerant varieties and the Vitis vinifera varieties in red, and find that the 2 genotypes do share the same metabolomic space, kind of denoting that although there are distinctions between all of these cultivars, largely they're all part of the same metabolomic fingerprint space. However, the caveat that they detected in this study that I find very interesting is that the anthocyanins in red wines were the distinction between the disease-tolerant varieties and the Vitis vinifera varieties. And this is because there is a unique anthocyanin profile in hybrids. So in Vitis vinifera, there's almost exclusively monoglucosidic anthocyanins. So we have our 3-ring flavonoid backbone and 1 glucose attached. And in hybrids, there are varying proportions of diglucosidic anthocyanins with our 2nd glucose attached. And this can have implications on the chemical matrix and the color of interspecific hybrid wines. So demonstrated here by the color of each circle, diglucosides are less colored at wine pH than their monoglucoside counterparts. They also are less reactive, largely due to steric hindrance, and they have a greater tendency to brown. Excuse me. So although there's not evidence of a negative influence of these compounds on wine quality, there is an OIV limit and a relatively low OIV limit for the content of malvidin diglucoside allowed in wines. Moreover, in Vitis vinifera, there is often a majority malvidin in the anthocyanin profile, and malvidin is based on its conjugation, a reddish purple anthocyanin. And demonstrated here in this nicely made figure are, thanks to Ananga et al., there are different hues represented by different anthocyanins based on the conjugation of the B ring. And in hybrids, we sometimes see large proportions of non-malvidin anthocyanins, which could potentially affect the final hue of the wine. So part of my research has been looking closely at a targeted fraction of anthocyanins in grapes and wine. And in this example, it is an anthocyanin fraction isolated using solid phase extraction, and the overall phenolics and anthocyanins have been extracted from grape skins using methanol. And this anthocyanin fraction was then, after isolation, analyzed via ultra-high pressure liquid chromatography. And here are Pinot Noir anthocyanin peaks in red, and which of course Pinot Noir is Vitis vinifera, and Vidocq peaks in black, which is a hybrid species. And these samples were sourced in France from a research facility that grew hybrid grapes because commercially hybrids can't be sold in France at this time. And Pinot Noir itself is unique among Vitis vinifera cultivars that has a very, very simple anthocyanin profile. It has 5 distinct monoglucosides, which are found in other Vitis vinifera grapes and wine, but it has no acylated anthocyanins. And here we actually see a very similar profile demonstrated by Vidocq. We have 5 distinct monoglucosidic anthocyanins and a few acylated anthocyanins. And part of my research has been to also explore how these anthocyanin profiles might relate to color representation. So this is represented by the CLAB color space. So that's the hybrid that is closest to Pinot Noir and to Vitis vinifera, but let's look at another hybrid, such as Chambourcin. So Chambourcin, I believe, is a Neo-American hybrid, which means it was bred focused on American species crossings and has been around for a long time. And what we find is that not only are there 2 big peaks for delphinidin monoglucoside and petunidin monoglucoside, there is a big peak for malvidin monoglucoside as well, but perhaps this ratio of delphinidin petunidin can have an impact on the color profile. There's also a sizable proportion of diglucoside anthocyanins, which are shown by these early eluting rounder peaks here. And so do we see a color difference? Yes, of course, because many different wines have many different colors, different grape types have different colors. But as we're looking at each of these different anthocyanin profiles, so shown here is a variety of them. All of these are different hybrids except for Pinot Noir on the far left. And as you can see, they all have very wide ratios of their proportions of monoglucoside anthocyanins, diglucoside anthocyanins, and then acylated anthocyanins includes cumarylated, acetylated, and caffeinated anthocyanins. And so with these wide, diverse anthocyanin profiles, what we're trying to elucidate is, do these unique anthocyanin features and these proportions of diglucosides have an impact on color expression? And if so, does that impact wine quality and consumer perception? And the other question is, are these wine colors still within the realm of the many hues and color intensities represented in the thousands of wine styles and cultivars around the world? So this is how we've been looking at the targeted subset of anthocyanins to explore these questions. And finally, this project is still underway, but I wanted to share a little bit about a non-targeted look at the chemical diversity of hybrids. So here we have 5 different interspecific hybrids shown in green. And again, these ones were sourced at the research facility in France. And then we have Vitis vinifera, includes Gamay, Gamay, and Pinot Noir from 3 different origins. And shown in this PCA plot is that what you could take away is that with this, I think there were 4,500 features or observations from the LCMS that went into creating these PCA plots. And so what you could take away is that if a point is closer to another, it means they're more different for simplicity's sake. And what we could see at first glance is that Gamma and Gamma Ray are relatively close to the hybrids and maybe even closer than they are to Pinot Noir. But that might also have to do with the difference of the composition of this Pinot Noir from Volnay. Regardless, what we did find was that there's a wide area to be explored within hybrids. So in the wine itself, there were 6,000 shared features between the Pinot Noir and the hybrids. And again, feature just is a single observation that came from the LCMS, whether it was annotated or unannotated. But we found almost 300 unique features that were in the hybrids that were not in Pinot Noir. And the skin extract actually was more complex than the wine itself, which was surprising. There were almost 650 unique features in the hybrids, not found in Pinot Noir. Excuse me. So overall, exploring the chemical fingerprint of these hybrids has shown that in some contexts, hybrids do have distinct metabolomic profiles. But as shown in the literature and starting to be explored here, the chemical diversity of hybrids is not or might not be dissimilar to the chemical diversity among the thousands of Vitis vinifera cultivars utilized around the world. And these takeaways can frame future research into hybrid grapes and wines, especially in my interest, and I think relative to real industry application, is that the connection between metabolomics and sensory research will be critical to fully understand hybrid grapes and wine. Excuse me, I'm so sorry that I'm sick today. Excuse me. So overall, I hope your takeaway today is that metabolomics has the capability to reveal complex chemical compositions, particularly in unstudied diverse cultivars like interspecific hybrids, which these compositions can then be applied to a variety of new explorations and applications, such as sensory perception, commercial application of hybrids, environmental response of the vines themselves, which I didn't get into at all, winemaking or aging treatments, and color reactions and color evolution and beyond. So I just want to say thank you again to the Group Polyphenol Committee for having me today, and I would be happy to answer any questions following the next talk. Thanks.
Thank you very much, Catherine. It was a great presentation.
Oh, thank you.
Fantastic presentation, Catherine. Thanks a lot for what you showed us today. So let's pass now to the second speaker of the day, is Nicolas Sommerer from Montpellier. So Nicolas got his degree in organic chemistry and biochemistry from the University of Toulouse. He got his PhD from the University of Dijon in food science. Then he entered a lot in the society of the French Young Mass Spectrometry Society that he served as a member, secretary, and also president. He worked also in Bruges as a specialist in mass spectrometry. And in 2000, he entered INRAE in Montpellier, that he started to work a lot in mass spectrometry, polyphenols, plants, proteomics, and so on. He passed different positions in his career. And in 2021, he was appointed as director of the polyphenols analysis facility of INRAE. And so, and today he's going to present to us next presentation about the chocolate color. So the title is From Light Brown to Deep Dark Chocolates, a Polyphenol Story. So Nicolas, thanks for presenting to us today. The floor is yours.
Thank you, Panagiotis. Thank you so much. Do you see my screen and the laser pointer? Okay, I'm making progress every day. my skills are maintained every day in webinars. So we will talk today of cocoas and polyphenols, and this study was made with a partner, which is Watrelot, which is a French high-quality chocolate maker. And we first, I will make some preliminary remarks to just remember everyone where chocolates come from. Chocolate comes from cocoa, as we all know, but the cocoa beans are fermented and dried before being shipped. So the product that we receive is cocoa bean, and all of this is produced in tropical countries. And the process is specific to the variety, the producing country, the producer know-how, and of course the harvest year. So this cocoa bean is shipped to northern countries and where they are transformed and consumed. There are many steps for transforming cocoa into chocolate, but the two main ones are illustrated there. The first one is roasting, which means heating at quite a whole temperatures, and then conching, which exposes during hours at intermediate temperature the future chocolate to oxygen, and this forms oxygen. In the world, fine cocoa represents nearly 12% of the total production, and the cost is quite high and quite benefit to the producer. What's on cocoa beans? Well, a lot of fat, of course, but also polyphenols. Nearly 15%, around 15% of polyphenols are present in cocoa beans, and well, we are going to focus on those polyphenols, of course. Well, this was a question. You see here 4 different chocolates, photographs of all different chocolates, and all those chocolates were made with 70% cocoa. That is to say that you have very black chocolates and very light brown chocolates. The brown one is not a milk chocolate. They are all made with 17% of cocoa. And as you can see on the screen, the colors are very different. So the question is, what are the compounds that are able to discriminate those chocolates? First, a very rapid outlook on the native polyphenols that are present in cocoa beans, either C-glycosylated or glycosylated compounds, B-type or A-type procyanidins. And on chocolates, of course, there are lots of exposure to heat, exposure to oxygen, and chemistry can take place. And of course, different kind of compounds can arise, and some of the compounds are colored compounds. For example, the A-type dihydrodicatechins are yellowish, and Thomas Hoffmann group in Germany show that xanthenocatechins and hydroxycatechinic acid radicals are reddish and can form larger reddish or brownish polymers when associated also with Maillard reaction products. So the methodology for this study was to transform the cocoa beans into chocolate with a standardized process. As to say, there's no process effect to generate the color. And then the color was measured within the LAB system, transformed into an image with the Adobe Photoshop image tool, and then a sensory analysis was made to sort the chocolates into 2 groups. The one that are brown and the one that are black. And we then did the experiment on those 2 groups. First, you have to defat, of course, with hexane, and then to extract the compound. And in our case, we wanted to extract many polyphenols, so we use a solvent appropriate for that. Go to high resolution mass spectrometry, and then a workflow up to identification thanks to our in-house complex polyphenol database. So here are the results. Well, very easily, you can separate with the PCA the black chocolates from the brown chocolates, okay? But you can also do that just with your eyes. What's more interesting is that this is a volcano plot, which is a feature that is very useful to compare two two sets of compounds. And in volcano plots, you see the discriminating features, that is to say, the discriminating compounds that are more discriminating black from brown and brown for black. And you see that on a total of about 371 features, there are 50 features discriminating brown chocolates and 26 discriminating black chocolate. I'm talking about features, not about compounds. So what is a feature? A feature is a signal in the mass spectrum, and which is characterized by an M over Z, its mass, by a retention time in chromatography, and the area of the peak. Okay, this is interesting, but this is not a molecule, it's not a compound. So how to go now to identification, to know all those spots, what are they, what compounds do they correspond to? I will just make a small slide to share with you one thing is that when you have an accurate experimental mass measurement, the software will propose you different possibilities of molecular formulas. This one seems to be nice with a very nice mass error, but you always have to check if this proposal is okay. And to do so, a very important tool is ultra-high resolution mass spectrometry. And that's what we did. If you zoom into the M+2 isotope, this is the 3 peaks that you can detect. The M+2 isotopes differs from the M+ isotope only by the presence of 2 additional neutrons. So as we have 2 additional neutrons, there we have 3 peaks. It means that those neutrons come from different elements, because the relativity theory said that intranuclear binding energy is mass, and there the difference in mass that we detect is the difference in intranuclear binding energy of the neutron. Okay, to be more clear, this is where the information is. This indicates us very clearly that we've got sulfur in our sample, because here you see the isotope 34 of sulfur instead of the isotope 32 of sulfur. There it indicates us the presence of oxygen also, as we see the oxygen 18th, and the same thing there with the presence of carbon 13. The difference between carbon 13 and carbon 12 is only 1 neutron. So in this case, you have to have 2 carbons to have the two mass differences. So with this tool, we are certain that this molecular formula is a good one, because we have the presence of the different elements, and we also have the relative ratio between the elements: sulfur to oxygen ratio, sulfur to carbon ratio, and oxygen to carbon ratio. Of course, having a molecular formula is not having an identification, so we have then to go to MSMS to identify the compounds. So I would like to warn everybody that with metabolomics, be careful when a database or a software gives you an identification. You have to check manually with MSMS spectra and with high resolution mass spectrometry that the proposal is appropriate. Okay, let's go now to a second tool, which is feature-based molecular networks. This is a tool that is very useful in complex polyphenol analysis, and the way it works is looking for fragment spectra similarities. As an example, you've got here 4 MSMS spectrum from 4 different parent ions, and you see very easily that some fragment masses are shared in between some MSMS spectra. But also some neutral losses are identical from one peak to another in the different spectra. And this allows the GNPS software to calculate a score, and the higher the score, the nearest to the number one, number one is the similarity higher. How do we represent that? We represent that in a network like this, and in this case, all those compounds have strong similarities in between them. The blue ones the same, and the same thing also for the yellowish one. We also have the information that there's similarities, but limited similarities between the green group and the blue group, and we also have the information that there's no similarities with those groups. Okay, this is theory. Let's go now to chocolate. So this is what we have on chocolate, and there we have an additional information. The additional information is the more red the spot is, the more present are those compounds in brown chocolates. The greener the spot is, the greener this compound is in black chocolate. For example, here you've got more green than red, and here you've got more red than green. So this is additional information that indicates you where does these features come from. Okay, how do we understand that? We understand that this is a cluster of compounds with large similarities, but for example, this one is quite different from this one, and this one makes some kind of subclusters, and so on and so on. And we have plenty of compounds there which are not in a cluster. They only resemble to themselves. They only look like to themselves, so they are not inside the cluster. But then there are some information into these compounds, and we will see this in the next slides. I would like to say that this is a partial view of discriminating compounds. Not all discriminating compounds are in a network. Some discriminating compounds are there in the lower part of the figure. Okay, a little bit too fast. What does this say? It says that in dark black chocolates, we have mainly O-glycosylated flavanol monomers and O-glycosylated A-type procyanidin dimers and trimers. And it indicates that in light brown chocolates, we have A-type procyanidin dimers and B-type procyanidins from trimers to heptamer. But again, not all discriminating compounds are in a network. If you go back to the volcano plot, you have some more compounds to identify, and the other compounds to identify are there. And so there are not only polyphenols that are discriminant compounds. You also have amino acid derivatives or oligopeptides, for example. Okay, to summarize very briefly this part of the talk, it's just to say that in black chocolate, we have small monomers or dimers that are O-glycosylated. And for dimers, there are A-type O-glycosylated dimers. For brown chocolate, you have much larger procyanidin dimers to trimers from B-type and to A-type procyanidin dimers. This is the main difference in between the two. Large one over there, O-glycosylated and small for black chocolates. So the question that arose now is that, okay, this is what we see from chocolate. What happens in cocoa beans? Because thanks to our partner Valrhona, we had also access to the cocoa beans that were used to make those chocolates. And when cutting the cocoa beans, we can see that they are light browns and also dark blood cocoa beans. That means that there's some kind of color pre-existing in the cocoa beans. And that also that we have seen when performing a phenolic extract, the one from cocoa bean is lighter than the one for brown cocoa bean is lighter than the color of the phenolic extract of dark cocoa bean. We did the same analysis and targeted metabolomics with nearly exactly the same workflow, just small changes in the extraction of polyphenols from beans from the beans, and this is what we get again. Very similar results with a very good separation of black and brown cocoa beans, and again the volcano plot with a little bit more features than with chocolate. And again, the question is, okay, what are those compounds? So the same tool, we use molecular networks, and this is what we have. In green, we have the discriminating compounds for black cocoa beans. They are over there in the network, and there's one there also in the network. And the red one, which are over there, and in this part, are discriminated for brown cocoa beans. And what are they? Well, for brown cocoa beans, they are mainly B-type procyanidins from dimers to octamers, and also A-type procyanidin trimer exoside. And for black cocoa beans, they are mainly monomers of catechin or epicatechin O pentoside or O hexoside. So if we want to compare the metabolomic profiles and the discriminating profiles between chocolate and cocoa beans, well, we can see that at least for the dark chocolates, well, they're the same compounds, or at least compounds from the same family. And when looking for the compounds discriminating for the light brown colors, well, again, it's nearly the same, apart from the C-glycosylated B-type procyanidin dimers and trimers that appear there. So can we say that those discriminating compounds in chocolate were pre-existing in cocoa beans? Yes, of course, but be careful. In some cases, there are different compounds appearing or modifications appearing. Because during chocolate process, I told you previously that we had high temperature, high time exposure to oxygen, and even if those compounds are seem similar, maybe we can't assay that there was some epimerization process in between the two. So they may be originating from that, or maybe have epimerization episodes in between them. Concerning brown chocolates, more things could be explained, for probably depolymerization process, and of course oxidation process to form A-type procyanidins and C-glycosylation. So there's the conclusion from that part of the talk is that there are strong similarities, but we cannot assert that similarity is totally independent of the process, because some new compounds form and some modifications appear. But they are pre-existing, the pre-existing metabolites were already present in the cocoa beans. The next part of the talk will focus on, well, stereoisomers. There are plenty of asymmetric carbons in polyphenols, and this may be a problem. And I will show you that this is a problem, but we have a solution. Okay, what happens if compounds isomer coelute in LC? Let's take an example. This one is a dimer. The extract an ion chromatograph of a dimer at a specific retention time, and this is the MSMS spectrum that we get. This MSMS spectrum, we can easily see that there are ions specific to B-type procyanidin dimers, but also fragments that are specific to dehydrodicatechin, type B dehydrodicatechin with a biphenyl interflavan linkage. So what does that mean? Well, this means that we have a composite MSMS spectrum. Okay, but this is a problem because when you want to discriminate the compounds, you may have false positive or false negative, because this signal is not a signal from just one single compound, but it's a signal for two compounds. And of course, when you perform the molecular network, it will be inaccurate because in this one, you will have similarities with B-type procyanidins, but also similarities with dehydrodicatechin. So how to overcome that? To overcome that, there's a tool, mass spec tool, which is called ion mobility spectrometry. Ion mobility spectrometry is just there. It's an instrument. The instrument we have is the Bruker one. It's a QTOF, traditional QTOF, high resolution mass spectrometer, but with ion mobility separation just over there. The principle of ion mobility is that if you have two ions, in this case it's two boats, which have exactly the same mass but different sizes. One being more larger and one being more folded. There's a nitrogen flow that pushes those ions into a slope of electrostatic energy, and they arrive to an equilibrium point where they concentrate, and there the equilibrium point depends on the collisional cross-section of the compound. The collisional cross-section is a projection in a 2D dimensional projection of the size of the molecule, but rotating in all the axes. And that's what we see there. So once you have accumulated the compounds, you have separated them into projected size, you can evaluate them by lowering the electrostatic potential. The larger one are eluted first, and the smaller one are eluted then. Okay, this is theory. This is what happened on polyphenols. So first, to understand what happened, we worked on a model solution. So this is not cocoa, this is not real life, this is a model solution of catechin and epicatechin mixed together with polyphenol oxidase, PPO, to form dimers, trimers, and so on. And we just decided to focus on dimers. So this is the chromatogram, extraction ion chromatogram of the dimers, and you see tens of compounds form during that polymerization. Okay. If we add the ion mobility separation, that's what we have. We see that for a specific peak, we have different spots. That is to say, there are plenty of coelutions. There, you've got at least 1, 2, 3, 4 spots. Then you got at least 2 spots. Then you got at least 1, 2, 3, 4, 5 spots. So this means that when you are looking for a feature in metabolomics, and when you say this peak, this extraction ion chromatogram has this kind of behavior, is overexpressed or underexpressed, well, you don't know what happens because there are several products behind it. So to understand a little bit more what's behind this, we first simplified the model solution and only using a catechin with polyphenol oxidase. And thanks to standards that we have, that are commercial for some of them and that we have in the lab, we're able to identify all the spots. We did the same things with epicatechin, and we're able to identify other spots, and lastly with the model solution. So you see that most of the spots are detected, but not all of them. This one, for example, well, we don't know what they are because we don't have the standards and we do not understand there. Okay, so this is great for molecular identification. This is great for identification, but what happens in real life? So the first thing we did, well, be careful, it's not cocoa. This is a grape seed extract. Well, we tried to use ion mobility, and okay, we have demonstrated in this paper that okay, they are very useful if you want to separate those compounds into natural samples. But grapeseed is not the point today. The point is cocoa. So we decided to go to metabolomics and to evaluate the added value of ion mobility. So we did the experiment with the same instrument with the ion mobility disabled or the ion mobility activated. The workflow is exactly the same. The only thing that changed is that we have two data treatments with the two sets of data. And this is what we get. Teams off and teams on. Okay, things look like quite similar, even if the PCA seem a little bit more clear with the teams on. And the second point is that we have more features with teams on than features with teams off. Okay. Of course, when making a molecular network, there are more nodes with teams on, as there are more MSMS spectra than with teams off. Okay, nice. But is it interesting? Let's check that. This is again an extracted ion chromatograph of a dimer, and this is the MSMS spectrum that we have without the team. If we turn the teams off, we see that we have 3 compounds. The mobilogram says, okay, 3 compounds are hiding below that, and so this MSMS spectrum can be divided into those 3 MSMS spectra. This one is characteristic of a B-type pyrosanidine dimer, and those 2 ones are characteristic of B-type dihydrodicatechin with the biphenyl interflavin link. Okay, this is again a great tool to identify sample, but is it useful in metabolomics? Well, the answer will be probably yes. This is the molecular network that we add without the IMS. So we see there one spot, which is discriminant for brown cocoa bean, but we don't know what's on that spot. Is it that one or is it that one? And that's quite important because this one doesn't have the same origin than this one. So if you want to understand what's happening, you have to understand which kind of molecule you have. So this is what we get with the teams on. You see that we have two clusters, and that the free ions are separated into two clusters. This one is alone, and those two ones are in the same cluster. And in this case, if you perform the discriminant analysis, you see that the B-type procyanidin dimer is discriminant, and this one, the B-type dehydrodicatechin, is discriminant, but this one is not. So in this case, we are discriminant compounds. We didn't know which one was discriminating. Well, and we have three compounds, but in those three compounds, just two of them are discriminating. I will go quite fast with the with other results. The same with flavanol dimers with two kinds of compounds. Okay, let's go again. What happens in this case? We've got a biphenyl ether interflavanic link. What happens into the molecular networks? Well, again the same. You've got one spot, one feature which is discriminant for brown cocoa-albin. Which one is it? We don't know, and if we push the teams on, we see that they are differentiated in two very different parts of the cluster. And among those four compounds, three of them are discriminant for brown cocoa beans. For flavanol trimers, we have to zoom a little bit more to see that there are B-type procyanidin primers and dehydro-3-epicatechin of type B. Okay, and again, if we do a molecular network, well, the compound is there, but it's not discriminant. With the teams off, this feature was not detected as discriminant. But if you turn the teams on, you see that you have one spot there, one spot there, one spot there, and one there. And those four features, among those four features, this one is discriminating for brown cocoa beans. And the B-type procyanidin trimers are not. The last example will be this one. This one is glycosylated flavanol monomers. Again, we've got two spots in the mobilogram. And when checking at the neutral losses, we think that there's a difference between an epicatechin O exoside and an epicatechin C exoside. And these are the results. This feature was discriminant for black cocoa beans, but which is it? And if we perform the analysis with teams on, the catechin or epicatechin O exoside is there in this cluster. And there's another cluster over there, which is with the epicatechin C exoside. And this one is discriminant for black cocoa bean. This one is not discriminant, but just remember what I showed you previously. It was discriminant for brown chocolates, not cocoa bean, but chocolate. So there's something that we have to look a little bit more in the next weeks. So to summarize, this was the cluster we asked with teams off and with 1, 2, 3, 4 type of interesting compounds, discriminating compounds. And this is what we get when we put the light, the light of the teams. You have more resolved molecular networks, and you have much more information on which compounds are discriminating and which compounds are not discriminating. The B-type procyanidin dimers that were there, you can have them over there, and the same for the other kind of biphenyl ether dimers, B-type procyanidin trimers and dehydro-3-catechin, and also for the O exoside and C exoside. You have just 4 information, and then you have, I didn't count, maybe 15 information. So the change of scale is absolutely tremendous. And that's what a part of exploring what we call the dark metabolome with ion mobility. And this dark metabolome is very huge when you are performing metabolomics on polyphenols, on dimers, on trimers, because they are not such small molecules than usually metabolomics focus on. We are looking on much higher compounds with lots of asymmetrical carbons. So there are a lot of heterogeneity and also a lot of transformation and change during the process, during the making of the final food, the chocolate in our case. So to finish with, the take-home message will be this one. thanks to ion mobility, we have improved the social molecular networks, and this highlight different differential isomeric markers. I would like to conclude that without ion mobility, you can see a lot of things, but you don't know exactly what you see. You are half blind. But with ion mobility, well, you still have difficulties in seeing very well, like this poor pirate, but you have a glass feed that allows you to see much more things. To conclude, I would like to thank my colleagues, and first of him will be Dr. Aisio Diaz, the postdoc in our lab. We did all of this great job and great efforts to have all those results. And I also would like to thank Dr. Véronique Chénier, that some of you may know. She's now retired for 2 years. But I remember that when 6 or 7 years ago, when I told her, Véronique, we should apply for funding to go to ion mobility, well, she wasn't that sure that it was a good idea, but she trusted me. And well, I would like really to thank her today, because I think she was right, and I hope that I convinced you that you are convinced also that it was a good idea to do so. So thank you very much, and I'll be happy to answer the questions. Thank you.
Okay, perfect, Nicolas. Excellent talk. I'm also a big fan of ion mobility, so yes, you did definitely a very good job, and it's really worth to go in this direction. So once again, I would like to thank both speakers. You can now use the chat box that we have to place all your questions. So any question that you have, you can write it there. We will see it, and we'll try to make these questions to the speakers. So now we have time for some discussion. So let's start with so we'll do this job both together with Aude. So we'll not mix up a lot. So let's start with Catherine. So Catherine, thanks again for your presentation. So the first question is mine. There are questions in the chat box, but the first question is mine, because I remember also like a PhD student in our laboratory that was working in very similar subject. So when she started, we were all saying that these hybrids have everything wrong. They have the wrong anthocyanins, they have the wrong aroma, they have the wrong tannins. And in the end of her PhD, we saw, maybe it is not like that. So did you arrive in a similar conclusion? So like, do they have everything wrong, these hybrids?
So sometimes I kind of get on my soapbox a little bit, because I wouldn't say like I think hybrids have everything perfect. I would not say that. But one thing that after working for this many years studying hybrids, I've learned that we often group them to get all together as like the other compared to Vitis vinifera, where what we're learning is that they have such diversity among the different types of hybrids, just like different cultivars in Vitis vinifera do. And so sometimes some of those, like you said, maybe bad aroma might exist in certain cultivars of hybrids, but not all of them. And then some of the unique colors exist in some of them, but not all of them, as I showed today. And so I think it does a disservice to the interest in hybrids to group them all together like that and say, oh, they all have these certain attributes, when what we're finding is they have diversities among themselves. And what I'm finding is that they even have kind of like, maybe you might taste two wines that you think, oh, the color and the flavors are similar, but not the same. In hybrids, it's the same. As you saw, Vidocq and Artaban were the two cultivars that are bred similarly, and they have similar parents. So they have many attributes that are close, but they still have distinct attributes for each one. So yes and no. It depends on the cultivar, is my takeaway.
That's a good answer, Catherine. I would add that there is nothing wrong or perfect, or it all depends on what you mean by everything is wrong. They are just so different. All right.
Okay, so have the next question.
Yeah, so we have a question. I think it's gonna go both for Catherine and Nicolas. So you mentioned about using C-Lab, so LAB system, to examine the relationship between color and anthocyanidins. C-Lab includes three-dimensional color space, but when you analyze the relationship with anthocyanidins, did you use hue value or did you employ another metric? So maybe more, Catherine.
So I could start, and then please feel free to take over. So for those of you that don't know, C-Lab is a three-dimensional color space, as this question asker said, where the L value is lightness, so light to dark, white to black, and then there's A star, which is red to green, and B star, which is blue to yellow. And this three-dimensional color space attempts to mimic how humans perceive color, and largely it's helpful in perceiving difference of color of two different samples. Answer the question of hue, I did use both the C-Lab color space where you can calculate A and B hue. It's just a ratio of over A, I believe. And we also used a more traditional method that's often used in the wine industry, using the absorbance at 420 and 520 and 620 with UV spectrophotometry in order to quantify color. And this is something I find really fascinating, is how we as humans try to numerically measure color when it's really challenging to do. Even ignoring all of the subjective aspects of color and what depends on the human perceiving it, very challenging to put a number to these interpretations. And so I was using all of these different metrics of C-Lab and their calculations and spectrophotometry and their calculations. And in the end, although you can compare between samples in one metric, you can't compare the hue by spectrophotometry to the hue calculated by C-Lab. So the answer is yes, we used a number of different metrics to look at how the color perception could compare to other factors. But there's not a perfect answer to like what is the defining color factor that people can take away.
Thank you. Nicolas, do you want to add something about LAB and the analysis?
In our case, the case is somewhat different because there are no anthocyanins in chocolate nor in cocoa beans. So the question is a little bit more different. And there's also a question from Nicolas Sommerer that we probably have to answer then. And so I will come back on that later.
Okay, I don't know if can I do a question to the next question to Nicolas? Or okay, so Nicolas, like Aude asked, do you have experience with compounds responsible for black color formation during alkalization of cocoa nibs to make black cocoa powder?
No, we don't have experience, and it's very difficult. This question was already a question by Thomas Hoffmann Group from Munich. He published in Journal of Agricultural and Food Chemistry. I can send the reference later in 2019. 2 articles with compounds that are precursors of colors, and mainly xanthenocatechins and hydroxycatechinic acid as radical ions. And those compounds, when they're polymerized, he showed in his publication that they form reddish to brownish pigments when they are of high mass, let's say superior to 10 kilodaltons. That's what they said in the publication. But what we also know is that there's of course association with Maillard reaction products with polyphenols. So the question of color is really complex. As you have mentioned, and as some of you have mentioned, when we are talking about flavanols, flavanols are colorless. They absorb at 280 nanometers. They don't have any color. So the idea was to see which compounds are discriminant for, but it's different than the question which compounds are responsible for. So the compounds responsible for are much more evolved compounds, probably bigger compounds, evolved compounds during oxidation process and combination probably with Maillard reaction products.
Thank you. And I think we have a question that is talking about Maillard reaction. So to keep talking about that topic, the question is about the procyanidin you showed are not colored, but probably are color precursors. Do you have a hypothesis for the real pigments? If you compare brown to dark beans, I assume it's due to extensive Maillard chemistry during fermentation with higher temperature. Would you agree? Lastly, for the networks, did you try as well the bet work feature in thermocompound discover? An excellent talk. Congratulations.
Okay.
It's a lot of different questions, but.
Thank you, Nikolai, for that question. So that was what I was saying. Of course, those compounds are probably precursors, and no, I think it's very difficult. We are not specialized on that, but it's really difficult to have access to these compounds, because these compounds are very large compounds. And when you are playing with mass spectrometry, we all know that going to high mass in mass spectrometry is difficult. We can do some nice things with proteins because there are plenty of nitrogen that are that can capture the proton and have a charge, but it's really difficult to charge them and to make them fly in the mass spectrometer. So we don't have this experience, but I guess it will be very complicated, and I guess it will be something very complex, because from one small break you add another break and you add another complexity, and that makes lots of compounds which global intensity is high but which individual intensity is very low. So you should have to check some kind of family group behavior to see the whole signal. That it will be very difficult. I think we are all convinced that there's no one molecule, one compound responsible for this color or this color, but it's plenty of molecules having very similar properties. And what we are probably able to do is try to understand what kind of compounds could be formed, but to have a full image of what compounds are really formed, I guess we won't be able to do so. And the second part of the question was using Compound Discoverer. Yes, we use Compound Discoverer with the Orbitrap instrument, but I don't know the bet work features. I don't know. I don't know that one, so so I don't use. Well, I don't know the bet work features in Compound Discoverer, so I'll have to check what it does. I don't know. But with the Orbitrap system, there's no ion mobility, so there's a big part of the information which is lost. We are blind.
Like, apparently one instrument is never enough, so we need more instrument possible to do all this job. So Catherine,
just a remark. That's a pity, because it means that if you want to do metabolomics, you need to have 2 or 3 very expensive instruments. So for lab managers, it's awful. Not very proud of that. For years and years, I said no. With a single instrument, small instrument, if you are understanding what you do in mass spectrometry, you can have superb and very nice results. Well, unfortunately, the things have changed a little bit, and now if you don't have the next, the best bazooka, well, the structure won't be relieved.
Yeah. Technology is important. Technology is important and helps a lot. So, Catherine, there is a question for you from Mujayan. I think so I say the name correctly. Like, which part of the grapes you analyzed? Did you analyze the seeds too? And if you can say something about that.
So within the scope of my PhD, I've had a few different sets of samples. One were commercial hybrid wines from around the United States. And the set of samples that was in France was, in my work, we only extracted from the grape skins and then pressed the juice, and then also received the wines from the same producers. So they were directly the same grapes that they made wine into. But at that time, we did separate the seeds and freeze them to hopefully be part of a future work. But even though 18 months seems like a long time, the visa was short. So I had to limit my scope a little bit. But yeah, it would be interesting to continue to work on that.
Great, thank you. I was looking at what questions we have. So still talking about fruits and grape cultivars, we have a question for you again, Catherine. So Anya is studying the formation of fruit quality. I have already read about sugar metabolomics of grape cultivars and implementation of metabolomics approach to study influence fruit thinning in grape cultivars. Thank you for your inspiring research. Have you combining your metabolomics research with transcriptomics approach to reveal the polyphenols metabolism pathway between the interspecific grape cultivars? So have you been focusing just on metabolomics, or have you thought about transcriptomics as well?
My work has, and the labs I have been working in have been largely only focused on metabolomics, and I think transcriptomics could be interesting, but in my understanding, metabolomics, because of its how do I put this? Like there's metabolomics and then transcriptomics and genomics that get smaller and smaller. And metabolomics is, to me, the best way to apply directly to whether it's the plant or the product itself because of its bigger molecules. But the transcriptomics would be an excellent way to zoom in on that polyphenol metabolism. But no, we haven't been able to dig into that. That's not the realm of these labs I've worked in, but it could be very interesting.
Okay, so Nicolas, I have a question for you. Like, I remember a few years ago, I realized that monomers, flavantriols, they can epimerize. So we have like we have and epicatechin and catechin, and they can also be created also. And sometimes we have them in cocoa. And I don't know if they can be formed during the high pH, when we have like high pH, this type of compounds. So I was wondering if this type of compounds we can separate, because they co-elute together with catechin, like catechin will elute together with epicatechin. And it's a pity with the method that we have for the moment, we cannot separate them. We need like a specific column to separate them. I don't know if you saw something with ion mobility, if it is possible to separate this type of compounds, and if you found any difference on these compounds between the dark and the light dark chocolates.
For the last part of the question, no, those compounds were not discriminating for light brown and deep black chocolates, but it's always a question of threshold. Let's say it's up to you to set the threshold where you want. When you have 400 features, you want to select, let's say, maximum 15 features for each kind of condition. Let's say brown versus black. If you lower the threshold, you will see more discriminating compounds, but of course with lower confidence. So it should be interesting to check whether these flavanol trioxyl are present. I haven't checked that, but yes, you can quite easily perform the analysis with an extracted ion chromatogram, and then check all the ions, and is they co-eluted. Again, use the ion mobility dimension to try to see whether they hide somewhere in the chromatogram. But we haven't tried.
Yeah, okay, thanks.
Thank you. So we have a question still for both of you. So what can be the strategies to bring more linked informations between metabolomics for hybrid grapes or oxidized structures in cocoa to process transferome in wine and chocolate? Kind of classification. Nicolas, you want to start?
Well, I guess I don't really understand what the question is. The question is to make a link between metabolomics and transferome.
Yes, potentially looking at more the process, how, yeah, transferome.
Okay, this is exactly what we want to do, because when I say that we are performing metabolomic studies, well, metabolomic studies on cocoa beans maybe, on chocolates not. Chocolate is not a living product. So chocolate, the evolution in chocolate is just process related. So we use metabolomic approaches, metabolomics tools, but this is not formally, and Catherine explained that very clearly. Metabolomics is not that. We use metabolomics tool, but to see different things. So what happens during transformation processes? And of course, the term metabolomics is not appropriate in what I've presented. Even for cocoa beans, because cocoa beans are already processed, they're already fermented, they're already dried, sun-dried. So this is not metabolomics. But this tool is really useful to understand what's happening and to point out the differences between the samples and between the process steps. So you can look the things between samples, and you can see the things between process steps. You can see the things in two dimensions. So and this is very complex. So to dig a little bit more into that, you have to understand more what's happening. Well, I guess that the next step should be to go into more small steps. In between cocoa and chocolate, there's grinding. Okay, what's happened during grinding? And then there's roasting. Okay, what's happened just after roasting? And then there's conching. So what's happened against? So you have to multiply the steps. And for cocoa beans, it's the same. You have to check genetics. You have to check the harvest year, so the vintage. You have to check the country, the producing country, and you have to check what the producers does, because in tropical countries, fermentation is raw fermentation in big boxes with large banana leaves, no starters. Fermentation is natural. They mix to have an homogenization in temperature every day or every 2 days, and then sun drying is in the air, and sometimes with birds, with chickens, with everyone on the same floor. So all those steps should be it may be interesting to characterize them, but it will be a tremendous work. Well, I don't know. For the moment, we don't have the chocolate partner to do that job. But okay, I will be interested in that.
I was just going to say that was an excellent answer, and I have a lot to add. I was thinking along the same lines of monitoring across the production cycle and seeing pinpointed things. And within hybrid grapes, we didn't have perfect control of production, and so there was a factor that would be included there, that we had different, perhaps slightly different production techniques across the board. And so in order to truly monitor that, we would need to control for that factor and look. So thank you for your answer, Nicolas.
Catherine, I have a question for you. Like you said about this limit that OIV has about malvidine the T-glucosides, 15 milligrams per liter in wine, that if you have that, the one is bad. Okay, the one is from a hybrid. So I want to ask you, do you know why like OIV decide to have this limit if the compounds are not really cause any problem?
So to answer that question, we have to dig into the history of wine, especially with the OIV and in France. And largely hybrids were originally bred to combat diseases, but like I said, when they first were bred back in the 1800s, they weren't bred for quality. And so there was a wave that almost all the vineyards were wiped out in France, and then hybrids started to be planted to combat diseases. And then they found that these hybrid grapes were not the quality that they wanted French wine to be. And I'm sorry if I'm pinpointing the French, this might have happened elsewhere in the world as well. And so now there are laws in France and with the OIV that essentially, like you said, the malvidine diglycoside is essentially just a marker for hybrids, because they found that most hybrids had malvidine diglycoside in them. And so if you were illegally blending your wine, then with hybrids, then it would mean that you were using these illegal grapes. So it's interesting to see, like for example, last summer at Macro Wine Conference, there was a lot of conversation and talks given about disease-resistant grapes. And as I showed, the OIV does have it part of the strategic plan, but as far as I know, I don't think the legislation of what can or can't be grown has changed in France. Although in other countries, there's growing all the time of what's allowed to be grown as part of and sold as wine, and that it's not necessarily bad. It's, even if it's a little different, it's been vetted to be qualified enough to make high quality wine. So I hope that answered your question. It has a lot to do with the history of where hybrids came from and how different they are now, but that we still have carryover of that origin.
Thanks.
Thank you, Catherine. A question for both of you, or for all of us, I guess. Do you think metabolomic approach is a good approach to develop tools, predictive models, to predict the stability of color in a final product? So thinking more about the grape and the wine, but that can be applied for other metrics. What do you think?
Should I start?
But
I think it's so challenging to the phrase to predict the stability of color is very challenging because that means that you know what the optimal stability could be and you can kind of predict around that in my eyes. And so I do think metabolomics could be a good way to look at the fingerprint of anthocyanins and their interactions in a wine and maybe see perhaps that browning might occur or how that color might progress. But when we talk about color stability, that is a big question of mine, is what makes a wine color stable and what is preferred by consumers when they taste a wine if it's aged. So yes and no, I think it could be interesting to look into that, but I don't know that even though metabolomics can give us this wide view of the metabolites and then we can compare that to color, in my experience, color perception itself is such a complex mixture of the anthocyanin fingerprint, so the distribution of different anthocyanins in the wine, the concentration of anthocyanins, and mostly the interaction that those anthocyanins have with other elements in the wine matrix. And so although, yes, metabolomics can be an excellent holistic view of those interactions and the fingerprint, I don't know that we can perfectly translate that to what the color will be or what the color will be in 10 years. That's an interesting idea. I would be curious to look into that.
thank you, Catherine. Nicolas, do you have anything to add for predictive models?
Predictive model, no, my answer will be the same as Catherine. We have worked on that, not me directly, but the lab on color stability for rosé wines, and well, again, metabolomics is able to predict the compounds that are responsible for stability, but then you have to monitor those compounds, and those compounds are in complex matrices, so they are, I guess, much more compound than only polyphenols to be able to predict. But it's maybe part of the answer.
Okay, Nicolas, I have one more question for you. Like from Hamitra Rabbeson, I think so I pronounced that correctly. So I work on protein polyphenols interaction. So it is easy to detect if this interaction is covalent or not. Thanks for your excellent talk. So it's about the interaction between polyphenols and proteins. What type of interaction is that?
Well, I'm not a specialist of polyphenol and protein interactions. Some other members in my lab are specialists of that. For chocolate and cocoa, the color is really stable, and I'm not sure that the quantity of proteins in chocolate is that important. I'm much sure that the quality, the stability of the color in chocolate is due to very complex compounds, polyphenols complex with Maillard reaction products, and I'm not sure that proteins do have an impact on that. And to have answers concerning protein polyphenol interactions on wine, for example, well, I don't have the answer because I'm not a specialist of that, but I can address you to some of my colleagues that will be able to answer that.
Thank you very much for all your questions. We have just 2 minutes remaining, so I just want to make sure we have time to, first of all, I would like to thank all of you for participating, but especially a big thanks for our speaker today, our fantastic speakers, Catherine and Nicolas. Thank you so much for your great presentation. Also, I would like to thank Dr. Arapitsas, so Panagiotis, for your great moderator session today. Also, in the background, you don't see them, but Jua-Pekka Salminen and Denis Baron, they are with us today. They are just managing the recording and making sure everything is going smoothly. So thanks very much to both of you as well. So just before closing, I would like to mention a few things. So the presentations are going to be available on YouTube, but also on the different websites. So the website of the group Polyphenols, as well as on Instagram, Twitter, a lot of different platforms where you will be able to find that recording in about a week. So give us just a little bit of time just to make sure we have time to process that. Also, you're going to receive an email in a few minutes that includes a link to a survey. So please take 2 to 5 minutes to complete that survey. If you don't have time, please scan the QR code that is right there. That's going to lead you to the survey. Also, before closing, I would like to mention that there is the international workshop on anthocyanins and betalains that's going to happen in September at the University of Leeds, UK. So if you want to submit an abstract, you still have time for a poster only. So the deadline is the end of July. So you still have less than a month to submit your abstract for a poster. Otherwise, feel free to join. You can scan the QR code that is also on the screen. And also stay tuned with us, because in a year, almost a year from now, we're going to be in Turku for the International Conference on Polyphenols. So Turku, Finland. So please join us next year in Turku to talk about polyphenols. You're going to learn a lot about that. On that note, thank you again very much for attending today, and have a good day. And I will see you later for the next webinar, potentially in November. So stay tuned with that. Thank you so much, everyone. Have a good day.